Alright, welcome back. We are on lesson 21.6, looking at protozoans this time. And so I'd like to take a moment and review some of the things we learned in the last lesson. We talked about different types of worms, and so there were three main groups. We had segmented worms, or annelids, flatworms, or platyhelminths, and then you had the nematodes, or the roundworms. Those were kind of the three different types of worms. Now, annelids, there were only three, so you had earthworms, sea worms, and leeches. There was quite a bit about earthworms, just in general. So they had those segments separated by septums, or septa. They had the setae, the little grabbers. They had aortic arches on their heart, 5 on each side. Or for their heart. They had the nephridia, which were kind of like their kidneys, and then they also had a couple other little things. Now, we also mentioned the sea worms. Remember that they had parapodia instead of setae, so they could kind of essentially breathe underwater. And of course, you've got your leeches that have the suckers. Once we got to flatworms, we talked about platyhelminths. Those particular ones we mentioned planarians. We mentioned the others, let's see, flukes and tapeworms as well. And then I essentially described kind of the lengthy process by which a fluke basically reproduces in fecal matter, uses a snail, comes back through grass into the other animals, and the particular worms basically live on and on and on. Now, tapeworms, remember they have that unique head called a scolex, and then we mentioned the different types of roundworms, or nematodes. And so we had filaria, which caused the elephantiasis disease, hookworm, which comes from the ground right into the foot of the host, trichinoworms, causing the trichinosis disease, and then also the ascaris worm that could affect your puppies. And so a couple little things there. Now, on page 445, we start by looking at the protozoans. Now, I understand at this point you've had a quiz over this material, but we are going to finish chapter 21 today together by looking at the single-celled creatures. And so starting off with protozoa. Now, protozoans, let me see if I can get this going. Protozoans are single-celled creatures. Everything we've talked about up to this point in the animal kingdom, if I can call it that way, have all been multicellular, even in the plant kingdom. So if you remember, Linnaeus originally said everything was animals and plants. As science advanced, we found out that there were actually more things, and these particular ones are single-celled creatures. And so these are unicellular animals in kingdom protista. Prot means one, and so you've got unicellular animals. Zo means zoo, or animal, and so one-celled animal, and that's what that's about. So most are microscopic. You wouldn't see these, and again, that's why Linnaeus obviously didn't know much about them. Thought of as simple, but are not. The more you study these intricate creatures, the more you'll learn. Each individual cell is a complete organism performing digestion, excretion, respiration, movement, and even responsiveness. These are more complex than most cells in the human body. Now, the reason I say that is we're saying these things are simply one cell, but if you think about your epidermal cells, although complicated, are not as maybe as amazing as these particular creatures. And you can find them almost everywhere. Alright, so there are four main groups, so I'm going to go ahead and do this, and it's based on their type of movement. So you have flagellates, or flagellates, number 2 sarcodines, number 3 ciliates, and number 4 sporozoans. Flagellates move with the flagella, or flagellum, singular. Sarcodines move with false legs, ciliates move with cilia, and sporozoans are sessile, so they don't usually move. Alright, let's start with those who move with flagella, the flagellates. Members of this genus, of the genus Euglena, are especially studied. So, of the flagellates, you have Euglena, and so Euglena are these green ones. If you look at page 446, there's a picture of a Euglena, and he's basically a jelly bean with a string on it. And so, looking further, these typically, if you do a sample of these, you get a pond water sample, pop it in the microscope, you'll probably, if you look hard enough in some greenery, find one of these, but it is pretty simple to find these. They're pretty common. What makes these unique compared to almost everything else is that they have chlorophyll. So not only do they eat normally, but if they don't have food normally, they can just eat with sunlight. Now, instead of this separates them from plants in the fact that they don't have a cell wall. If you notice the picture on page 446, instead of having a cell wall or a cell membrane, they actually a thickened membrane called a pellicle that allows it to basically be elastic and move. We'll talk about kind of the euglenoid movement in just a second. They move in two ways. First of all, they use a flagellum. Flagellum's a whip-like tail that they use, and then of course they use what's called euglenoid movement. Now, this one is almost more like how worms move. They actually can change the shape of their body. It's an incredibly important thing. It's unique to euglena and how they move, but this movement that they can use by changing their shape. So be familiar with that. Again, videos would be very helpful if you get a moment to pop through and see some of those. All right. Well, here's a euglena. I don't actually think this is one. I think someone just put some jelly. This looks like it's from a fishing lure or something, but a little string off of it. Essentially, that's all you're looking for. Now, this stigma, this red dot here, is actually the eye spot, but it's a good model for us. And so let me mention a couple things. First of all, the pellicle is the outside edge. Got a nucleus in the inside. You got chloroplast for photosynthesis, and the stigma or eye spot is very often used for for keeping it moving towards light and such. All right, moving on a little bit more with euglena. There's so much to say about these that basically near the flagella is an opening. Now notice it says interior rather than posterior, so you would think this is the back of it, like it's the tail or a motor on a boat. It's not. It's actually the front of the animal. Of course, it opens into the gullet. That's kind of its esophagus. It has a lot of these contractile vacuoles. These are vacuoles or food storage chambers. Vacuoles just do that. They store food and nutrients. Of course, they have a nucleus. They possess chloroplast. We mentioned that. And here's your red photosensitive eye spot. Now, many times books will call it the stigma of the euglena, but that's what we're looking at. Now, obviously, these things reproduce by asexual reproduction. They actually just split, that binary fission. I'm sorry, that's a typo right there. That should say the word fission. And so binary fusion is something totally different. And so let's see if I can find there we go. So typo, let's put fission. Okay. Fusion puts things together, fission separates the two. And so they reproduce by binary fission asexually, so they simply just split into two, and each one gets a new, basically gets a new nucleus and genetic materials. And so that's, again, how that kind of looks. Again, notice the stigma, the chloroplasts that are clearly obvious throughout this creature, and even the splitting here. All right, they mentioned trypanosomes, or trypanosomes, as your book pronunciation says. In this genus, and so I will go through quickly, but basically they're parasitic ones. They also have flagella, but they actually have two, transmitted by the tsetse fly. This is what causes African sleeping sickness. Basically feeds on blood, undulate membranes, so they basically move like manta rays. Here's your flagella, and they basically influence your blood. All right, here's your tsetse fly that transmits those. Sarcodines, these are protozoa with pseudopods. And so we're on page 446 looking at those protozoans with pseudopods. Remember, they're named by how they move. And so flagellas, or flagellates, are going to be those who have flagella or flagellum. And so you have sarcodines that are going to be amoebas. Specifically, that's kind of our main example. And so I will go through three, it looks like, but then I will be done with these. Amoeba is perhaps the best known and most common of these. They have flexible bodies that can shape at will. Those flexible bodies really are a result of these pseudopods or false feet. Basically, the amoebas can change their shape through these pseudopods, that pseudo, false, pod feet. And so they can basically engulf things with those and eat. Eats by engulfing particles, engulfing them in a vacuole. So basically, they will surround it with a vacuole and then start to digest, similarly to coelenterate. They also, notice I spelled that right this time, fission. They also undergo binary fission. So here is an amoeba. You'll notice all of its organelles inside, and also these pseudopods. These are the fancy things that give it the name sarcodine. Now, a couple more things to be said. No eye spots. So you'll notice these things have no way of seeing. However, they do in a, excuse me, they do in a microscope seem to have some form of response to bright lights, and so we can't really explain at this point. Science doesn't understand how they move based on that, but interesting. They can't have no eye spots, but they do see light, or respond to it at least, obtained by collecting slime from the bottom of a pond. So you want to get a euglena, just get you a pond sample with tons of plant matter in it. You'll be able to find it. You want to find an amoeba, get the slime at the bottom. You probably will find them. And really, that's a lot of fun. You'll see a lot of really clear creatures roaming around in there. Obviously, one of the diseases that can be transmitted by an amoeba is this amoebic dysentery. And so just be aware of that. Now, there are two other sarcodines that I wanted to mention, and those are foraminiferans, that's how you say that, foraminiferans, and radiolarians. And so those are simple to look at. So they have these calcium carbonate shells, whereas radiolarians have silica shells. Now, sarcodines, typically the amoeba has kind of like a jelly shell, no shell at all, if that makes sense. And then foraminiferans, these two actually do, but they are made up of different things. Here are your foraminiferans. They kind of look like corals, but they are in fact more similar to an amoeba. And then, of course, your radiolarians. Man, there's just so much design in this. Look at these cool shapes that you can see kind of everywhere, even at the microscopic level. And this guy, this was Boppet before Boppet ever became a thing. All right, we're looking at 447, talking about ciliates. And so we've talked about really two already. We've been kind of fast. We've been looking at euglena, which were flagellates, or flagellates. I'm trying to say that word right, with obviously avoiding to say the silly version of that. And so ciliates are protozoans with cilia. Cilia are kind of like a conveyor around them. The best example of this is the paramecia, or simply singular there, paramecium. Well, if you look at a paramecium, you would notice it looks kind of like a slipper. Before I go further, that's not really as helpful, but you can definitely see the shoe or the foot shape of this particular creature. And if you look around it, you'll notice the little blur that can be seen inside of this creature, or the outside of it. Those are the little cilia, the strings around it. Actually, the book says they're actually submarine-shaped, but in cross-section as seen in a microscope, which is how you're always going to see them, is going to be more of a slipper shape. Now, if you want to collect weeds in a pond within a few days and let them grow, you'll find more paramecia in there. Now, they actually have two nuclei. They have a macronucleus, big, and micro, small. Now, their body is divided into two layers, ectoplasm, endoplasm, and they also have a pellicle, which is not their cell wall, but their stretchy outside. So again, here's kind of our special material that we're talking about. Well, we'll come back to these trichocysts. Those are coming back here. The gullet, of course, this is how they eat. Anal pore, cilia, these are going to be the portions. It kind of looks like a conveyor belt or an army tank in the microscopic world, but of course you got your macronucleus and you have a micronucleus, and then these super cool, and they actually are star-shaped contractile vacuoles, and so it's food storage chambers. Very cool. So there they are, paramecia with their cilia. Now, embedded in the ectoplasm, or the outside layer of it, are these things called trichocysts, which are dart-like structures that are shot out when attacked. So when I say this thing is a tank, I'm not kidding. Apparently they can put out these things that look like almost fiberglass needles. These are called, thank you Fine Art America, but these things are called trichocysts. These are like spears that this thing can throw out. Now obviously dart-like structures, I got these two lines mixed up, food vacuoles and contractile vacuoles if they need to eat. Now these are very important because they're the #1 consumer of algae. They're actually kind of like vacuum cleaners of the pond. And of course they have this oral groove, resembles a funnel. Food is forced into it by the cilia. Going back to the picture I had just a minute ago, all the food, if you kind of look at these little hairs, everything gets conveyed by the conveyor inside of this oral groove. Everything gets pulled in. And so that oral groove resembles a funnel. Food is forced into the gullet by cilia. Liquid wastes are excreted by these two star-shaped contractile vacuoles. And of course, undigested food is released by an anal pore. Okay, and so again, here's kind of your picture. Phagocytosis, that's how you say that, is the eating of cells. And so these things do eat very small things. So food's getting pulled in by the cilia into the oral groove, the gullet, of course, going further in. They'll form a vacuole around that and digest it. Again, one more picture. Notice the shoe shape of this thing, the paramecium in general. Kind of fun. All right. Paramecia do not have nerves but can respond as well, similar. They reproduce in two ways, and fission is one we've talked about, but also conjugation. That's going to be seen on page 448. If you want to read through that, it's pretty intricate, actually. So here's basically what I got. In conjugation, two paramecia exchange nuclear materials, then they both divide repeatedly until you've got a bunch of different paramecia. So conjugation is kind of their form of splitting. Notice in fission you have two creatures. In conjugation you have quite a few, quite rapidly. So that's what we're talking about. I don't know that this picture necessarily because right now we don't know the term meiosis, we don't necessarily know diploid, haploid, but we will come back to this concept in later chapters when we have a better understanding of the terms. A couple others that I wanted to mention. Your book kind of lists them off as Didinium, Vorticella, and Stentor. Didinium is a barrel-shaped ciliate that often feeds on paramecia, so he's kind of the bad guy here. You'll see a picture top left of 449 of a Didinium. In fact, I have one here. Didinium is kind of barrel-shaped in the sense that it's rounded. You see the cilia, which give it the name ciliates up here. But again, kind of a gullet up here. But this is Didinium. Vorticella. My brain says vortex. Maybe that's not helpful. Maybe it is. It looks more like an upside-down bell. It's a stalked ciliate, so it's going to be attached to something commonly found in stagnant ponds. And so here's Vorticella. Looks kind of like this. It looks like an upside-down bell. You kind of got your stalk that goes up, and then of course your bell. These are attached to kind of algae. Then Stentor is trumpet-shaped that can be found swimming in fresh and salt water. So Stentor is very unique. When they say trumpet-shaped, they mean it. You can see the cilia at the very top here that kind of projects outward. You can see the little chloroplasts and different things. Here's a clump of algae down at the bottom. But if you ever do pond samples, you may see one of these three, as well as Euglena, even paramecia. And so you want to be familiar with what kind of little creatures you can run into. All right, let's move on to page 449 and start talking about the sporozoans. Sporozoans, just making sure, okay, where we're at in this particular slide. And so sporozoans are non-motile protozoans. Non-motile, they don't move. And so they're spore forming. That's why they're called sporozoans. Almost all parasites that require two hosts. They're transmitted from one host to another by mosquitoes, flies, ticks. Asexual and sexual reproduction, they're responsible for the disease malaria. Specifically, they undergo something called the alternation of generations because they exist in multiple forms. And then, of course, you've got Plasmodium. It says it has one of the most complicated light reproductive cycles of any creature. Plasmodium is the one that causes malaria. And so kind of, to be honest with you, this is probably the most simple picture I can find online. Again, it's going into different forms. Here's a cycle within a cycle. Within a mosquito, it's different than inside of a human. And so this is that alternation of generations. But sporozoans, again, very tiny, not really something you can show a picture of, but are a thing. All right, and that concludes our chapter. And so tomorrow, your proctor will be showing you quite a few lab videos of just dissections of some of the various creatures we've been talking about in this chapter 21. When I get back on Monday, we'll start chapter 22 on cytology and really kind of head our way towards the end of the year. Thank you for your time.